Bonjour et bienvenue to the third episode of Active Concepts Masterclass Season 2. I'm Raphael Tron, Marketing Manager for Europe, and today we are going to discover another phase of our cells with the study of cellular senescence. Let's start to see what cellular senescence is about and how it could be linked and tested regarding aging. And for that, I will have the help of our Manager of Clinical Validation and Toxicological Assessment, Daniel Schill, who you have met in episode 1 on dermal microbiome immunology assay, and if not, you should definitely see that video afterwards. For most of us, our appearance and physical attractiveness are significant factors in how we communicate and interact with others. So not only they impact us biologically, but also psychologically, shaping our overall identity and influencing our confidence and emotional well-being. The anti-aging market has grown significantly with an estimated worth of 60 billion according to Global Newswire. And by 2030, it's expected to increase up to 120 billion. However, despite these amazing figures, current anti-aging solutions appear not really satisfying for customer expectations. The latest trend towards well-aging further emphasize this. And so as an alternative, senolytic ingredients may provide a solution. Hi Dan! Hope you are doing great and that you are ready to dive into the senolytic world. So today, with our episode, we are going to discover our senescence-associated beta-galactosidase test performed on keratinocyte model. Wow, what a name! Dan, could you maybe explain first the overall target of this test?
Hi, Raph. Thanks. I'm doing great. I'm happy to help assist in understanding this assay model. So mainly, we developed a cellular aging model to assess the in vitro effect of our ingredients to reduce the senescence process by measuring SA beta-gal in aged keratinocytes.
I think it would be better maybe to first define some terms in order to better understand what is happening. Then you talk about a model of cellular aging. Why should we use this principle?
Yeah, sure. So what is important to keep in mind is that skin aging is a clear indication of the gradual transformation of the body, which is a natural and inevitable biological process. So cellular and molecular biology is one of the disciplines that can explain many of the mechanisms responsible for skin aging. So the goal here is to artificially recreate aging cells in order to be able to test our materials and see their impact.
Okay, so I understand that the start of the test was to simulate aging, but how does this happen?
Great question. Yeah. So to simulate aging, we use keratinocytes, which are harvested and reseeded in cell culture flasks. These are called passages, and the number refers to a simulated age. So low passage keratinocytes present morphological, metabolic, and senescent biomarker profiles corresponding to those of young skin in vivo. And then conversely, high passage keratinocytes present in vivo the cellular characteristics of older skin.
Perfect, I understand the system well, but why start from here? What does this ultimately represent for aged cells?
So as we age, DNA damage accumulates and leads to skin cell dysfunction and death. Cells that do not die continue to function poorly. And so the biochemical and molecular foundations of aging are rooted in the mechanisms of progressive failure of homeostasis, which corresponds to a statue of a state of balance. And this ultimately results in the accumulation of damage in the nucleic acids, proteins, and lipids.
So the cells no longer function properly, is that right?
Correct, yes. So this results in an alteration of functional capacities at all levels of the body, which increases the risks of various diseases. So indeed, the homeostasis of a living system depends mainly on its maintenance and repair processes. So thus, the progressive failure of these mechanisms constitutes the universal biological basis of aging and age-related diseases.
I understand that trying to fight aging can allow us to be in a better shape and have healthier cells. But ultimately, what process is specifically studied here?
All right, so during this test, the goal is to see how our ingredients act on cellular senescence.
Okay, but what exactly is it?
Great question. So senescence is the general term used to describe the biological changes that occur as an organism ages beyond maturity. So one notable aspect of this process is cellular senescence, which involves normal cells losing their ability to divide while maintaining activity.
Yes, it's true that cells have the ability to divide to promote the growth, the maintenance, and repair of cells and tissues. Finally, they no longer divide but remain in a somewhat suspended state. But what if they can no longer divide? What happens?
Right, so the major disadvantage of senescent cells, in addition to their inability to contribute to tissue maintenance, are their harmful effects on neighboring cells. So these cells release harmful factors like pro-inflammatory mediators, molecules that damage the extracellular matrix, oxidants, and then several other molecules. So these are characterized by changes within the cell morphology, metabolism, and signaling pathways, and biochemical profiles.
I've heard of zombie cells regarding this phenomenon. Can you tell me a little bit more?
Yeah, so as we've said, senescent cells will secrete a complex of factors, also known as the senescence-associated secretory phenotype, or SASP. So continued exposure to SASP can transform normal cells into senescent cells. So they gradually become contaminated and become more malignant, hence the link with zombies.
Okay, in fact, over time, as a result, these elements begin to degrade the structure of the skin, which manifests itself, for example, in an increased reduction in collagen, wrinkles, and thinner skin. Ultimately, this concerns the visible signs that we want to combat as we age.
Exactly.
So, for our test, we start from aged cells, but what are we specifically studying on this test? Are we trying to measure certain molecules released by cells, as you mentioned to us just before?
Right. So the SASP cocktail released during the senescence process notably includes a typical biomarker called senescence-associated beta-galactosidase, or more simply SA-beta-gal. It is the reference biomarker for identifying senescence in vitro because the enzyme beta-galactosidase explicitly accumulates in the lysosomes of senescent cells. So SA-beta-gal expression is strongly correlated with cellular aging in cultured cells.
So we will compare young cells and old cells to measure the quantity of SA-beta-gal and see how our ingredients can prevent its formation?
Right. So in this assay, the young keratinocytes are low passages and aged keratinocytes are high passage cells, but they're from the same batch and used in the same culture plate and then cultured to confluence. So these young and old keratinocytes are incubated with only the growth medium as a reference control. And then we test our different product concentrations on the aged keratinocytes.
Okay, I understand the comparison. But once incubated, how do we identify the quantity of SA-beta-gal?
So for this, we use different cell permeable dyes. So we used Hoechst and Spider beta-gal. So these two dyes are used in a ratio to provide an indication of senescence in the cells. So the Hoechst dye fluoresces brightly when bound to nuclear DNA to indicate cell nuclei, and then Spider beta-gal fluoresces when bound to beta-galactosidase, indicating cellular senescence.
Why make a ratio?
So a ratio of the two dyes is necessary so that we can control for any differences in cell count that might occur between the young and aged keratinocytes. So ultimately, SA-beta-gal activity is normalized by taking the ratio of the beta-galactosidase signal from the Spider beta-gal to the nuclear signal from the Hoechst dye. Then we set this ratio relative to young keratinocytes to highlight the effect of cellular aging on senescence.
Okay, wonderful. Thanks for that explanation on why we need to make this ratio to have the SA-beta-gal activity. I think it's really important to keep in mind that our goal is to have the lowest activity as possible, trying to reach the young keratinocyte activity to reverse aging process and reducing the secretion of senescence cells presence. This test has a strong link with the idea we highlighted at the beginning. Consumers are a bit tired of the usual anti-wrinkles test. They are looking for more and deeper benefits. And the raise of the wellness awareness and overall well-being really triggered the consumers to try to heal from their deeper side. Having a strong look on the intrinsic stress and cellular behavior towards the study of cellular senescence could help feeling younger and more confident. Thank you very much, Dan, for your help on understanding the importance behind our AC beta-gal assay for our masterclass series. So it was Dan, Manager of Clinical Validation and Toxicological Assessment for our US lab, and Raphael from our European marketing team. Do not forget to watch previous episodes and be ready for our next one about hair shaft elongation.